Historically, the Great Lakes food webs were relatively simple. And if we were to use Lake Michigan as an example, there was a top predator, and that was lake trout. And then those top predators fed on some smaller fish. That group was dominated by something called ciscoes.
The cisco were some of the most numerous and populous fish of the Great Lakes. They're really, really important. Cisco are sort of this perfect food source for apex predators.
So the ciscoes were comprised of 8 different forms. And so you can imagine that one ancestral cisco being in this ecosystem 12,000 years ago, and then over the course of thousands of years, it sort of split off.
Since then, humans have impacted the Great Lakes through industrialization. And so now we've seen a loss of cisco species diversity in the lakes.
Then the question is, well, what was the role of that diversity? And you could imagine that diversity, having more species, allows the system to be more stable and more resilient. These fish, if we're thinking about historical food webs, they don't exist anymore. We can't go out and collect them and sample them. But so we're working from museum specimens that have been preserved in museum collections for about 100 years. We go to these museum specimens, we take a tiny tissue sample, we take these chemical measurements, and then we make these inferences about the diet and the feeding of these species.
So from a piece of muscle tissue of the fish, we can measure the stable isotopes of carbon-13 and nitrogen-15, and that gives us a really, really good idea of where they're fitting in the food web. Because what you eat is reflected in your isotope signatures.
On the morphological side of things, we're going to use an approach called geometric morphometrics.
More recent advances in morphometrics is what I'm specifically interested in. So instead of calipers, I just take a photo of the fish. So you splay the fish out on a styrofoam board, you take a photo, and then there's a bunch of processing that goes on the computer where I basically place what we call landmarks on the same spot on every fish. So the tip of the jaw, the eye, insertion points of the fins. And so you place all these landmarks and you're essentially digitizing the fish. Our hope is that when we run our analyses that we can see sort of clusters of shapes, different clusters of fish that look similar, and then we can compare, do their isotope signatures also cluster? So these very similar looking fish, are they also eating very similar foods? There's a ton of field work involved in this study. Lake Superior is slightly more pristine, and so they have more intact cisco diversity. So that's sort of almost like your control. That's where the cisco diversity is highest. So we have fish coming from Lake Ontario, Lake Michigan, Lake Huron, Lake Superior.
But I think ecological restoration is sort of a growing movement. A lot of interest has been focused towards trying to bring back the fish that historically fed higher trophic levels, in other words, the ciscoes. And there have been some reintroductions already, and there very well may be more reintroductions in the future. So we want to maximize our understanding of the past and hopefully use that as a guide for moving forward.
It takes a lot of collaboration. I think Seagrant is an example of one of those great collaborative networks and is essential to any, I think, Great Lakes study.